So the next speaker is Ruben Dries, who will talk about the GIOTTO software suite. So let me pass it off to Ruben.
Right. All right, thank you so much for the invitation. I'm really excited to be here. And also thank you, Ron, for giving a beautiful introduction to why we actually developed GIOTTO Suite, which is a spatial multiscale and multi-omics analysis framework. All right, so we're going to talk about multiple different data sets and what we can do with it. Some of them have multiple different features. Some work at multiple different scales, or how can we leverage some of the existing spatial geostatistics frameworks that already exist to leverage or to increase our resolution for Visium data sets, for example. So GIOTTO Suite is the main topic of today, for my talk at least. So GIOTTO Suite is a suite of different R packages. And if you would like to know a little bit more information after this talk, because I'm only going to briefly touch a few key topics, then I invite you to visit our website, which is giottosuite.com. If you're a new user, I would definitely tell you to look at the examples, at the tutorials, and follow the news page, because we regularly add new ideas. If you're a developer and want to contribute, then definitely also check out our GIOTTO ecosystem, the different class structures that we make available, and how you can potentially contribute. All right, before I start, this is really a massive project that we have been working on for multiple years with many different people. It's a collaboration between my lab at BU and GC's lab, at GC Wen's lab at Mount Sinai. In my lab, the lead developer is George, and in the other lab, it's Jocelyn, and together they get a lot of support from other people in the lab and also funding sources. All right, so what are we trying to do? And I really need to say thank you to Anna Carpenter and Shantanu Singh, who recently put out this beautiful, very small papers about bringing computation to biology by bridging the last mile. And I'm just going to read out this thing out loud. So it's all about making usable tools that are the last mile bridge between what computer science makes possible, so what method developers do, and what biologists are able to put to widespread use in their research. So we're trying to make something that is as easy to use, but also as flexible to use. And that's not always that easy. All right, so why are we doing this? So there are many different spatial technologies, and you saw that in the previous talk. So we definitely need a technology agnostic structures and approach. You don't necessarily have to reinvent the wheel every single time there's a new technology. At the same time, the number of data sets and methods are exponentially increasing. So it's really now or never. And also, like, spatial omics data is complex. It's a lot more complex than your typical IF or your typical single cell. So it also requires more complex data structures, especially if we think about combining multiple technologies as well. Right, so here I'm just giving you a few of these, like what I call next generation data sets. One of them is from Tenex, where on multiple different sections of a breast cancer tissue, they apply their Xenium, their Visium, they apply IF. Another similar group, another similar paper, I should say, is made by a group in Belgium, where they also have multiple sections of the same tumor. They apply different types of spatial technologies. Again, something similar is where someone compare a group, they combine MERFISH and electron microscopy. Really exciting work. That what Ron Fan basically just described, basically doing the same, or on the same tissue slide, he was able to do epigenome and transcriptome co-profiling. Other groups are also starting to work on 3D data sets. So you basically make a 3D tissue data sets rather than a 2D data set. All right, so in order to basically analyze all of that and also think about how can you integrate these different technologies with each other, we're working at least on two different fronts. On one end, we're developing the software. So we're doing software engineering, where we think about how should we represent these data in the most efficient and memory efficient manner. Thinking about point data, shape data, the networks, how can you integrate that? And that is really necessary if you want to think about more like flexible and scalable spatial data analysis. Right. So how did we develop GIOTTO? What was like the original, the starting point? We looked at biology, and you probably all do, and we noticed that biology is definitely a multi-hierarchical layer, right? You have like your larger units or your larger tissue structures, and then you have smaller structures. You have your single cells. Within the single cells, you also have organelles, and within these organelles, you can identify all types of different profiles, molecular analytes, transcripts. This can be proteins. This can be anything you would like to. The technology kind of follows along, and the technology allows you to profile some of these molecular analytes, whether that's your protein, your transcripts, at different types of resolutions, as you saw previously, going all the way from micro subcellular transcripts to 55 micron and even bigger. So, and these technologies also represent, their output is different. So this can be your typical image. Think about H&E image, but also can be like individual points. It can be intensities, can be shapes. And at some point, you would also like to integrate all of that information, going all the way from your genetics, epigenetics into kind of like a vertical integrated connectome that defines your phenotype of your cell or cells of interest. So if we can have that all together, what we can do is we can literally profile any type of spatial technology that is available. And also and this is like a subset of some of the data sets that we have profiled, but we can also think about combining them in a kind of like a vertical manner in the future. So the key point that we do is, and this is about one of the only slides that talk about the software engineering point of view, is that we separate the features from the spatial units. And so what I mean with that is we don't necessarily combine them at the beginning, but we basically separate them. And the features can be any type of spatial modalities. And we have specific data structures to represent the points, intensities, to polygons, the networks as well. While the spatial units is a very loose conceptual idea that can literally be anything. And you can have like one spatial unit, or you can have 1,000 spatial units. It doesn't really matter. That spatial unit can be cell segmentations, can be any type of segmentations, can also be an array or some type of tessellation, spatial binning approach, or even you can start thinking about nested spatial queries. And at some point, you can aggregate these features and these spatial information in any type of way that you want. And you can do that 1 time, you can do that 1,000 times. You can do that across different type of technologies. You have multiple technologies, and that allows you to profile and integrate that data quite efficiently. All right, so I'm going to give you a few examples that just to show you what you can do and what you can build on top of that. And I'm also going to show you a little bit about how that we can do more like community and tool building using this specific framework or technology. So I'm going to start with a very simple multi-scale idea and what we can do using our kind of like spatial unit concept. So this is a MERFISH data set from a breast cancer data set. Breast cancer data set, for which we have information about the nuclei. So that would be one spatial unit, but we also have that for the cells. And so what we need to do for MERFISH, we need to assign these individual transcripts to a nuclei or to a cell. But we can also identify larger structures, right? For example, we can identify niches, we can identify domains, and then they have some kind of like hierarchical relationship, which we can visualize. So we can visualize how that kind of like goes, where you can go from domain, niche, to cell, and nucleus. But you can do a lot more. So you can extract that information, and this is a very straightforward analysis, and you can identify, for example, you can assign the individual transcripts to the whole cell. You can cluster that, you can identify the different cell types that are available. Now, you could do exactly the same thing with the nuclei only and assign these nuclei or assign the transcripts to the nucleus. And in this way, what you're doing is a spatial in silico single cell RNA-seq versus single nuclei RNA-seq. And then you might ask yourself a very simple question. Do you see that these clusters, do they match 1-to-1? Is basically the cell state, is that the same as the nuclear state? And in this case, it's clearly that's not the case. And that gives you a lot of like new hypotheses about what's going on. In the same way as your typical velocity is kind of like thinking about in which direction is a cell going. And another example, and I'm just highlighting some key examples, is thinking about registration and segmentation. 2 key concepts that are not that easy to implement, and they're also like a little bit unsolved in the whole field. All right, so on the one hand, now we're using the 10x genomics dataset, where they profiled xenium. They did an IF and they did Visium on adjacent sections of the same breast cancer dataset. And what we can do, and we use the tools from Gene Fence Group, ST-Align, to co-register the different datasets so that they have in the same framework, and then we import them into GIOTTO. We're also making some tools that we can do this all in R, making it a little bit easier for people to do this in the future. So once we have that data set, I think one of the key things in any of these spatial subcellular transcriptomic data sets is how exactly what kind of like segmentation should you use, and how is that segmentation important in the output that you're looking at. So what we're doing here is we're comparing the original segmentation. So these are the cell boundaries, basically, that are identified by 10x himself, and we compare that with Bezier. So in our case, these are like two different spatial units that you can have in the same GIOTTO object, and you can use them together to do a joint profiling or a joint clustering of these two different segmentation approaches. And ask yourself the question, do I see difference in cell type composition, spatial differences in cell type composition between the original and Bezier? So what we noticed doing this kind of like joint clustering is that the stromal and cancer-associated fibroblasts have a 100% increase using the Bezier specific algorithm. And you might ask yourself, is that actually useful or relevant? I think it definitely is, since we know that these cancer-associated fibroblasts, they are key to understand why a breast cancer tumor, where a tumor basically responds to treatment or not, and how it progresses to a more aggressive state. Staying with the same idea, we're now going to go and move a little bit into multimodal, and that what you can do on multimodal level. And we're simply going to compare, since we have this now integrated within a framework, we're going to compare modalities or across technologies. So we can compare modalities, for example, with protein versus RNA, and they only have, at the point that we analyze this data set, they only had like two different IF. They had one for CD20 and one for HER2, and then you can compare the protein level and the RNA level, and basically look at spatial correlation, and you see that it can be either like okay, or sometimes it can be a little bit better. A more interesting analysis is that you can now start comparing across different technologies and think about, do they actually agree with each other, or what is like happening? So here we're looking at genes that we find back in both xenium and visium. And we ask ourselves, do they spatially correlate? And what you see here, and this is for all the genes on the x-axis, that some of the genes have a very high correlation. For example, this one, FASN, but others do not really have that high correlation. And we were a little bit wondered why that was the case. And now we noticed that it's actually very nicely correlated itself with the absolute expression levels of xenium and visium. So overall, they seem to agree pretty well, except that when you don't have anything to work with, you're really working in the kind of like the noise levels, right? One other thing that you can easily do once you have that GIOTTO object established is you can combine multiple modalities with each other. So rather than just like analyzing everything on the RNA or protein level, and this is something that you could, for example, do with the spatial site-seq and what Rongfen introduced, or some of the other methods. So you can combine your RNA or protein network at some point using a weighted nearest neighbor network analysis, or anything that it's like that you would like to do, and combine them and have cluster results or your downstream analysis using both RNA and protein in one way or the other. All right, and scalability. Scalability, I think, is a really one of the key or fundamental issues that this field is facing right now, simply for the fact that the data sets we're working with right now are relatively small, and now people are generating really large data sets where they're trying to compare condition A versus condition B and so on, and it's becoming really a challenge. To illustrate what we're trying to do to alleviate some of that, at least, is we use the StereoSeq data set where they profile at subcellular resolution using sequencing data, a whole mouse embryo in development. So it's a massive data set. It's very hard to work with. So it's really, we're tackling this in many different ways, but I'm not going to go over everything in detail, but we do a lot of code optimization. We implemented some clever spatial chunking approaches. So you can like do spatial type of analysis in a chunk-wise manner, so you don't use that much memory. You can project your data, you can subset your data, make it a smaller data set, do your PCA UMAP or cluster annotations, and project it in the bigger data set. That's especially useful to kind of like explore your data set before you want to run it on the whole data set. And we also provide GIOTTO in the cloud, for example, on the Terra server, if you don't have your own infrastructure and want to scale it up. But I guess another interesting approach is basically using or making use of our very flexible tiling mechanism, right? Where you can do this and you can start the analysis on a much coarser resolution, for example, this hexbin 400, or you can do that on a hexbin 100. And so what it allows you to do is you can start with a very coarse type of like aggregation identify your region of interest. Let's say you're interested in that specific region. So that's all very doable on your local machine. So you don't have to go to a server. You get that information. Hey, that's an interesting region. Now I want to know a little bit more about what exactly can I find out in this specific region of interest. You can then simply create a new, much higher resolved hex bin. This, for example, hex bin 50. We already start to see a lot more of these layered structures that are available within the brain. You can apply all these different tools that we make available in GIOTTO to do other type of spatial data aggregations and clustering. For example, if you use the niche composition clustering, you really nicely see the very nicely beautiful layered structure within the brain, mouse developing brain. All right, just to highlight some of the innovation within our flexible tiling approach or a spatial unit way that we approach these things, you can also now all of a sudden from that specific data set, you can make another layer where you create a pseudo Visium data set. So what you do is you create your spatial unit becomes one of these like 55 micron spots. You can aggregate your transcript within this 55 micron spot. And now you created your own pseudo Visium data set. You might ask like, why do you want to do that? But maybe you have additional Visium data set. And you would like to see like with my specific tissue, which deconvolution approach works the best. So now you can use this to test your specific deconvolution approach that you would like to do using your single cell data or non-single cell data. Identify the different cell types, and then you can immediately compare it with the ground truth, which was provided by another technology, in this case, StereoSeq. All right. All right, so now the last part is I would like to highlight some of the community and tool building. So first of all, GIOTTO does not live in isolation, but we do try to be as interoperable as possible with some of the other common ecosystems that are available. And we do make it as simple as possible for others to add their methods or tools to GIOTTO if they would like so. But what I'm probably most excited about is that we build on top of known existing structures in the R spatial ecosystem. So the R spatial ecosystem is literally decades old, and they have a beautiful packages that do all sort of really amazing spatial statistical work. We make our GIOTTO points and polygons, which are these lower level data structures in our GIOTTO object. You can immediately convert them into spat vectors, SF, and stars, which is this objects that are being used by these packages that are very well established in the field and that have a bunch of different tools available that you can immediately apply. So I think if this is what I always show, if I have seen further than others, it's by standing on the shoulders of these giants. And these guys are really giants, right? If you see the number of different tools and applications that they have available. So if you can leverage that, you can apply that in your spatial data, you can all of a sudden do a lot more than you can do with any type of existing spatial data analysis platforms. So just to give you an example, something we built in just a matter of weeks using some of that is here on the right, you see a typical mouse brain gene expression profile for Panther1 that we analyzed with GIOTTO Suite. So, but there is also an associated H&E image, and that's associated H&E image. You can identify the nuclei if you have a good H&E image at least. And for this one, we use StarDist. So now we have another spatial unit. We have the spatial unit, which is the individual, the spots from Visium, but we also have the individual nuclei, which you can even expand a little bit. On the other hand, we're making use of the fact that we have this accessibility to the R spatial ecosystem and do something what is called spatial interpolation or kriging, which is a way that you can predict expression of genes or expression of specific features or variables in regions for which you don't have information, but if you think there is spatial variability or there's spatial covariance for your specific feature of interest, you can use that to build this type of idea, which people also use in geostatistics, and they use it, for example, to predict the amount of rain that is accumulated somewhere for where there is no weather station. But long story short, what you can go is you can go from something like this to something like that, where now you have predicted values in individual nuclei rather than individual spots. So you can do that for all the genes that have a very specific spatial expression pattern. Now we did that for 500 genes and use that as input for GIOTTO. So now you're using images as input, but what you can do is you can go to something like this, where you cluster your spatial patterns in a very typical way to something like this, where you don't look at the individual spots, but you're actually looking at individual nuclei. So it's much better representing the actual spatial architecture of your tissue of interest, at least for the spatial expression level. Last but not least, if you want to know a little bit more, I invite you to read our paper. I would also like to say that this is, if you do test something out and there is a bug, then please let us know. We're always interested to know how we can improve or what we can do to make it better. If you're interested to contribute, to add your tool, or to collaborate, then let me know, reach out, and happy to answer any questions after the session. Thank you very much.